Viruses and evolution, lifting the veil. Evolution is a fact according to evolutionists, and the common descent model is their smoking gun. Everything in biology falls into place when you go to the common descent model with the single common ancestor for all life, as opposed to the multiple ancestor or separate ancestry model, which is not parsimonious, not unifying. It's polyphyletic, many ancestors, as opposed to the common descent model, which is monophyletic. So the common descent model is a fundamental argument, a fundamental model for evolutionary theory. That's our background. Let's get into viruses. One of the most powerful evidences for evolution today is from viruses, and specifically viruses in our genome from birth. Endogenous retroviruses, and it's the pattern, they say. It's the pattern that makes evolution a fact.
In contrast, if we want to believe the fixed species view, we're forced to conclude that these viruses are simply shared by coincidence. When we do the math, even making sure to account for the 9 viruses not shared by the 2 species, the chance of this happening by coincidence is less than 1 in this crazy number right here. This evidence should be enough for even the most reluctant yet rational person to carefully set aside the fixed species view.
1 chance in 10 to the 1,418. Wow, that's impressive. That is a 1 followed by thousands of zeros, as you saw in the video. But this number actually has nothing to do with evolution. It is a deception. I will explain that in a minute, but first, viruses are crucial to evolution, more crucial than you might think. This paper says that evolutionists must consider scenarios of virus origins in conjunction with models for the origin and early evolution of cells. This dramatically ups the ante for the study of virus evolution, bringing it to the center stage of evolutionary biology. The center stage of evolutionary biology. Virus evolution really counts. Viruses are crucial to evolution. Here's a paper. Viruses are an intrinsic part of life as we know it, and accordingly, the origin of life encompasses the origin of viruses. Viruses are crucial to evolution. Another paper, evolution of life cannot be understood without elucidating the origins of viruses. Viruses are crucial to evolution. Yet these origins currently remain mysterious. So how did viruses evolve? Well, no one actually knows. One idea is that viruses evolved from a primordial genetic pool. Primordial. So viruses evolved prior to their hosts. Well, that's strange because viruses require their hosts. By definition, viruses require their hosts. So how could viruses have evolved prior to their hosts? Well, that is what some evolutionists argue for because the evidence does not fit the traditional idea of viruses evolving after their hosts. Another awkward conclusion is that viruses do not share a common ancestor. Quote, the conclusion seems inevitable that viruses comprise several distinct lines of descent. Comparative genomics provides no evidence of a monophyletic origin of all viruses. Many virus groups simply share no common genes, effectively ruling out any conventional notion of common origin. Several lineages of viruses probably evolved independently before the last universal common ancestor. So viruses evolving before their hosts and with no common ancestor? The science is contradicting the theory. Now, 3 broad ideas have emerged to explain the evolution of viruses. First, there is the virus first hypothesis, also known as the primordial virus world hypothesis. Now, we saw that a minute ago. Then there is the regression hypothesis, where viruses are degenerated cells. And then finally, there is the escape hypothesis, where viruses evolved from genes that have escaped from cellular life. It is widely accepted that viruses are polyphyletic, but a consensus scenario for the ultimate origin is still lacking. Traditionally, 3 scenarios for the origin of viruses have been considered but none of the three ideas worked. These classical scenarios give different timelines for the origins of viruses and do not explain the provenance of the two key functional modules that are responsible for viral genome replication and virion morphogenesis. The science contradicts the theory. So here are those three major scenarios, and not only have none of them worked, but they all call upon non-empirical entities. The first one calls upon replicative elements to be in that primordial world. Well, those have never been observed. The second one calls for the degeneration of cells that then assume a parasitic lifestyle. Well, that's never been observed. The third one proposes that cellular genes acquired the ability for selfish replication and spread. Well, that's never been observed. Virus evolution hypotheses call upon non-empirical entities. The science contradicts the theory. Over the years, all three scenarios have been revised, elaborated, temporarily abandoned, and revisited. So here is a graphic showing, illustrating kind of a cartoon showing these three different hypotheses, three different categories of scenarios. And one idea is, well, they don't work, so how about combining them? Here we outline a chimeric scenario, which is an amalgam of the primordial and escaped genes scenarios. So this is reminiscent of the DNA code theories, many theories, none of which work, so they try combining them. So if you can't get anything that works, how about if we put them together? Kind of a cafeteria approach where you pick and choose whenever you need one or the other. When one seems to work, you use it, and then it doesn't work later, so you pick some other one. So to summarize, viruses do not fall into a common descent pattern, and they defy evolutionary explanation at this kind of cartoon level. Initially, 4 vast realms of viruses were established, each including an enormous diversity of viruses, and then later 2 much smaller realms were subsequently added. The 4 major realms encompass the vast majority of currently known viruses, although they emerged independently. Independent, so not common descent. They're dropping common descent. So you had that unifying phylogeny. It was so important, that unifying idea, the parsimonious idea of common descent that was so important to evolutionary theory. It did not work with viruses. They had to switch to the polyphyletic pattern, the separate ancestry pattern, multiple virus ancestors, polyphyletic. The science contradicts the theory. But even with the polyphyletic model, multiple virus ancestor model, evolutionists cannot explain virus evolution. As we have seen, there are several different hypotheses to explain virus evolution because none of them work. When you have multiple hypotheses, sometimes it's because none of them are working. Now, there is a whole nother problem here, another bigger problem. So consider the viral cycle. So how do viruses work? You have these infectious virus particles called virions. They infect a cell, they take over, they hijack the machinery, and create a whole bunch more copies of themselves. The cell dies, and all these new copies, new virions, are produced. So that's the cycle. Virions into the cell, more virions. A competent virion is a very complicated molecular machine. A competent virion must somehow evolve by random causes. That's how evolution works. So how does the virion evolve? There is no plausible explanation. So let's look at an example from some images taken via transmission electron microscopy. These are really cool images. Now this is of a giant virus called virus. It was the first of the giant viruses to be discovered. And here is an image 8 hours after infection of a cell. That black line at the lower left is 5 micrometers. So you see the cell, and in the cell there is this black blob. It's labeled VF for virus factory. So this virus has infected the cell, set up shop, taken over, hijacked the machinery, and created this virus factory. And then you see these kind of hexagonal blobs surrounding the virus factory. Those are newly minted viruses being formed, coming out of the virus factory and and being completed. So you can just kind of see these new viruses being created in the cell. Zoom in just a little bit here, and now it's at 12 hours. That black line at the lower right is 3 micrometers. You see the virus factory there, kind of in the middle, and then a lot more newly minted viruses hanging around, waiting to spring into action when the cell is broken down. And here's some more images zooming in here. Look at all these newly minted viruses hanging around the edge of the virus factory being completed. Zooming in even more and zooming in even more, you can see these. And then finally we reach a beautiful hexagon, nearly completed Mimi virus. So it's an amazing and incredibly complex process of this, injecting into the cell and taking it over and hijacking the machinery. There is no plausible explanation for the evolution of these viruses. You will search in vain for a scientific, detailed explanation of how viruses actually evolve. Paper after paper after paper on virus evolution, they don't give you a scientific explanation for how these actually evolve. So viruses do not fall into a common descent pattern as we saw, thus defying evolutionary explanation, and the random origin of a virion also defies evolutionary explanation. No one actually knows how viruses could have evolved. Although several hypotheses have been recently proposed to explain the origin of viruses, it remains unexplained. The science contradicts the theory. Because of the great diversity among viruses, biologists have struggled with how to classify these entities and how to relate them to the conventional tree of life. To date, no clear explanation for the origins of viruses exists. The ultimate origins of viruses has proven to be a more intractable problem. What the primordial world looked like is open to debate, and there are a number of rather different versions of the pre-Luca theory, last universal common ancestor, pre-Luca theory. More striking is that most, about 70%, Mimi virus genes have no known homologs in either virus or cellular genomes, so that their origins are unknown. The actual mechanisms leading to the emergence of sophisticated parasites that are both able to exploit cellular resources and to mediate their own transfer into a new host cell is far from being understood. Yes, understatement. Naturally, our results are obtained from simulations with a limited complexity, and thus, as with all evolutionary models, caution needs to be exercised when they are applied to the real world or to actual events that took place on early Earth. Yes, well said. Evolutionary studies are far from realistic. The general scenarios for the evolution of the virosphere remains a matter of intense debate. Evolution of life cannot be understood without elucidating the origins of viruses, yet these origins currently remain mysterious. In any legitimate taxonomy, all taxa must be monophyletic, which in the case of viruses presents a major problem. There is not a single gene common to all viruses. Therefore, viruses are polyphyletic, and accordingly, a unifying phylogeny of all viruses is impossible to obtain. Now, here's a highly produced, fancy review paper in a leading journal on the origins of viruses, specifically retroviruses, endogenous retroviruses, origins and evolutionary consequences of ancient endogenous retroviruses. Very highly produced paper. So if there was ever a paper that was going to tell us how these endogenous retroviruses evolved, it would be here, right? In fact, it says right here, this review describes ERV origins. Oh, well, wonderful. We're going to learn about this. Well, the paper doesn't. You can search in vain through this paper. Here's a quote. Thus, ERVs are genetic loci whose ultimate origins trace back to exogenously replicating retroviruses. Well, okay, that's how they transition to the endogenous retrovirus. This isn't the ultimate origins. This is nothing about how they evolved. Likewise, here's another quote. ERVs originate as integrated proviruses and can range from complete proviruses to highly fragmented remnants of proviruses. Yeah, okay, this is the process they go through. This is not the ultimate origin. This is not the evolution origin of ERVs. The science contradicts the theory. So to summarize, virus evolution is crucial. Without it, evolution cannot be explained. Evolutionists have no detailed step-by-step explanation for the origin of any single virus. Evolutionists do not even have broad cartoon-level scenarios describing virus evolution at a high level. There are many attempts, none of which work very well, so it is not for lack of trying. Evolutionists admit virus evolution is a big unknown. Evolutionists believe viruses evolved not because of the science, but in spite of the science. So this is very important. Evolutionists believe viruses evolved because they believe evolution is true. No one actually knows how viruses could have evolved. Virus evolution is contradicted by the empirical evidence. The belief that evolution is true underwrites the assumption that viruses must have evolved. If you're going to argue that viruses are powerful proofs of evolution, you might want to mention that viruses contradict evolution in the first place, right? There is no plausible cause for the evolution of viruses. Now, I pointed this out in a presentation about endogenous retroviruses, and evolutionists disagreed, but he did not address this problem. He did, however, respond to another problem, mismatches between the LTR and proviral body sequences. So what's that about? Let's look at that. LTR versus proviral body mismatches. This is another enormous problem with the idea that viruses evolved. So first, the architecture of the virus. For a virus that's going to become an endogenous retrovirus, it has a protein coat, and then inside there is the genome with protein coding sequences and flanking regulatory sequences. So this is going to be injected into a host cell and then ultimately integrated into the host genome through a very complicated sequence of events. So what's injected into the host DNA, the host genome, is called the provirus. And you have that proviral body, the central part, which is those protein coding genes. So there's 4 protein coding genes coding for a whole bunch of proteins. And then some flanking sequences, which are called long terminal repeats. So what we saw in the virion, those regulatory sequences, those are processed and become what's known as LTRs, long terminal repeats, LTRs. At the integration event, these 2 LTRs are identical. Now, what about the question of when this integration event occurred? Evolutionists have several methods to estimate the dating of that integration event. So, several different methods to use. Well, the LTRs in the proviral body contradict evolution in just about every way possible. These integration dates that we discussed, they're often conflicting. Phylogenies or evolutionary trees derived from these sequences are often conflicting. And even across the genome of the same organism, they will conflict. And I'll show you some examples of this. So they contradict the theory. Here are some examples. So here's a paper that's looking at a particular endogenous retrovirus in humans. The proviral body and the LTRs are what? Phylogenetically different. Clearly, proviral bodies appear much more homogeneous in sequence than the associated LTR sequences. Now, why in the world would that be? Under evolution, LTRs must have evolved independently from their proviral bodies. And since purifying selection makes no sense, the reasons for this are currently not clear. So that's a problem. Here's another paper. Now this paper is from the same group a year later, and they're part of that paper. They're talking about the same endogenous retrovirus and going over that. Proviral body and LTRs result from different evolutions. Under evolution, quote, there is presently no explanation for the significantly different sequence alterations in the LTRs versus the proviral bodies. Now, here's another endogenous retrovirus they looked at, and it showed very different behavior across the genome. So in the Y chromosome, the LTRs had a much greater divergence from each other, 16, almost 17% divergence, and that would indicate an integration date of at least 64 million years. Whereas on the other chromosomes, this endogenous retrovirus, the LTRs had only about 10% LTR divergence, indicating about 40 million years for the dating of the integration event. So they had to say that the problem lies with the Y chromosome ERVs because the species distribution was too narrow for the 64 million year age. So they had to say that the Y chromosome LTR divergence, those data were misleading. But how to explain the divergence? Why was there this LTR divergence on the Y chromosome and only on the Y chromosome? This is strange. So what they said was there were a lot more mutations that accidentally accumulated in the LTRs of a provirus on the Y chromosome. Now, there were a dozen of them on the Y chromosome. So what's going on there? Well, they said there was a single provirus that had accidentally more mutations, and then it amplified in copy number on the Y chromosome. So a circuitous sequence of events they had to call upon, but even still, there was no explanation for why a provirus would sustain so many more mutations in its LTRs, but not the proviral body. Well, it was an accident, so that's your explanation. Okay, let's look at another paper here. There are many examples of low LTR-LTR divergence. So here the LTRs are pretty similar. That would indicate a recent integration event. So in this case, less than 2.5 million years ago. Now, the human-chimp speciation event, the human-chimp divergence, was thought to be something like 5 or 6 million years ago. So at less than 2.5 million years, these should be only in the human lineage. You shouldn't find these in the chimp, right? But about half of them did have homologs in the chimp. So that makes no sense on evolutionary theory. Another glaring problem. And then there was this. Actually, the majority in this particular family, the majority have a discrepantly low LTR divergence. They just don't make sense compared to the proviral body. Okay, let's look at another paper. Now in this paper, there are two ERV families thought to be closely related. So pretty similar, kind of similar proviral body. Those protein coding genes were somewhat similar. So they must have come from a common ancestor sometime back in history because these genomes were similar. But not so with the LTR sequences. They were much more different. Again, this mismatch, LTR proviral body mismatch. But there is a whole nother problem going on here. So we just saw that there was a mismatch between the LTRs and the proviral body. We've seen plenty of examples like that. But here the dating is also a problem because both the proviral body and the LTRs, the divergence calls for an older date than the species distribution implies. So the species distribution requires or indicates a dating of less than 60 million years ago. Okay, but the LTR and proviral body divergences require much more time. Now, these divergences I'm talking about are one of the two ERV families. Remember, we're talking about these two ERV families that are thought to be related by common descent. They took one of them and compared it to a template consensus sequence and looked at how much it had diverged from that consensus sequence. And both the LTR and the proviral body had diverged quite a bit. Now, the LTR much more so, much more so. The proviral body not that much, but even then, the proviral body as well as the LTR required a dating much older than the 60 million years. So again, another mismatch. So we've got the mismatch between the LTR and the proviral body, but also more dating mismatches going on. So a lot of problems in this particular example. So how can this be explained? Well, they say it is in line with previous observations. So there are a lot of other observations like this. A lot of other mismatches have been observed like this. And in other words, don't worry about this LTR proviral body mismatch because it happens commonly. I guess if I have a theory that is contradicted by the evidence, then it's okay if it happens a lot. That's kind of how this works. Okay, let's summarize this LTR mismatch problem we've been talking about. I won't go through this slide. You can pause it if you want because I just went through all these details. But this is kind of a summary of what we just looked at. There's just, as I said earlier, the evidence from the LTRs and the proviral bodies contradict evolution in just about every way you can imagine. And these are glaring contradictions. They do not work under the theory. The science contradicts the theory. Now, let's look at our evolutionist friend and what he said about this. Recall, this is the one where he addressed this and said, hey, wait a minute, this is not a problem. So what did he say? Let me just summarize what he said here. He said it's okay because this LTR proviral body mismatch, it's okay because as protein coding genes, the proviral body sequence could be under purifying selection and thus more conserved. So there's an explanation for why the proviral body had less divergence than the LTRs, and you have this mismatch. The proviral body is under purifying selection. These proteins are crucial. They're vital to the operation of the endogenous retrovirus, so they couldn't sustain these mutations. It was under selection, purifying selection. On the other hand, as regulatory sequences, the LTRs would not be. And they could, or in addition to sustaining, more easily sustaining mutations, they could actually be adapting under positive selection. So there's a couple of explanations for how you could have this sort of a mismatch. And he says that this is a plausible reason. And he says that this is known. These sorts of trends are known, plausible and known. Well, no, none of this is plausible, and no, none of this is known. So this is not a good explanation, and it is circular. But let's step back for a moment before we get into that and just take a quick look at this.
Right? So if you were to go on Google Scholar or something, right, and you were to look up bat wing evolution, how do wings evolve? How did whales evolve to be marine? You're going to find literal books answering those questions.
This is a good example of how evolutionists think. Now, I have a video on this, and you can see the link below. First and foremost, evolutionists believe evolution is true. Evolution must be true. It is a fact that cannot be questioned. So the bar is very low. They do not reckon with the actual science. Thus, discourse tends to be incommensurate, what philosophers call incommensurate. They are perfectly comfortable with speculation, just-so stories, non-empirical, unobserved causes and mechanisms, and circular reasoning. So discourse is incommensurate. You're talking about science. They have a belief. They have a belief in a theory that cannot be shaken. Science, it is robust. It is not vulnerable to science. Now, in this case, everything the evolutionist is saying is either circular or ignores major problems. So back to this explanation that our evolutionist friend gives here, and recall the dating mismatch. So there is a big dating mismatch we looked at. His purifying selection idea, his purifying selection explanation aggravates the dating mismatch that we looked at. It aggravates it. It makes it even worse. He is going in the wrong direction. This is not working. He also has the wrong omega trend. Under purifying selection, they would expect greater sequence difference at the nucleotide level than the amino acid level for these, the proviral body, but the paper notes no such trend. Actually, ERVs are typically disabled. Yes, there are proteins that are occasionally functional, such as the syncytians in the placenta. We'll look at that in a second here. But that is the exception, not the rule. His claim that ERV proteins are under purifying selection because they are vital for the virus ignores the fact that ERVs typically have long since become disabled. So there's going to be no purifying selection. Now, his arguments are circular. No, regulatory sequences are not known to evolve more rapidly relative to protein coding sequences. They're not known, as he says, as he claims. That is an evolutionary inference. Evolutionary inferences are not axiomatic. Now, this is, again, back to this incommensurability problem, where evolutionists will very commonly and easily hold these evolutionary inferences to be facts, because after all, evolution is a fact. So those become part of the argument. You cannot use them in defense of evolution. That is circular. It's the same problem with the hidden Markov model paper that he cites at the end of that section there. That paper also, that HMM paper also incorporates epigenetics, makes an appeal to epigenetics. Well, this is a problem. Not only is the evolution of epigenetics unlikely, but evolutionists bitterly opposed anything resembling epigenetics for over a century until they no longer could deny it. And now our evolutionist cites this paper to argue for evolution. So this is circular. This is a train wreck. The LTR proviral body mismatches contradict evolution. The science contradicts the theory. Okay, let's move on to another glaring problem, and that would be that endogenous retroviruses enable host function. Here is a paper about the placenta, mammalian reproduction. ERVs provide, quote, a major driving force in the emergence and diversity of the placenta.
There's an amazing story about one of the genes in these endogenous retroviruses that's used by humans on a daily basis, and that is a protein called syncytin. Syncytin is an essential protein for making the outer layer of cells in the placenta. It's formed by a single layer of fused cells. These are all syncytia, right? Fused cells with many nuclei and one long tube of cytoplasm. And that makes the maternal-fetal barrier, right? There's fetal blood there's maternal blood, and the syncytia trophoblasts are an essential part of that barrier. The reason they fuse is because they have a protein called syncytin, which is derived from the envelope protein of an endogenous retrovirus. So remember, when retroviruses bind to cells, they have an envelope protein that binds a receptor, and then the membrane of the virus fuses with that of the cell. It's catalyzed by the glycoprotein. It's a fusion protein. So the gene encoding that, it's called envelope, has been taken from the endogenous retrovirus and put somewhere else in the genome, and there it gave rise to the placenta. This happened millions of years ago. It happened 3 different times in mammalian evolution. Mammals have placentas, right? But there are some older mammals that do not have placentas, like marsupials and before them. Capture of syncytin 3 times has led to the development of a placenta in all of these species. So you can see syncytin 2 here gave rise to placentas in many of all of these species. I just noticed this, old word monkeys. They use ancient English. And then another integration gave rise to gibbons, orangutans, gorilla, chimps, and humans. So it's a gene taken from an endogenous retrovirus. So this probably happens a lot, and this is the best studied example. So this is why we give live birth as humans, because we have a placenta the baby can develop in utero for months and then come out quite well developed, as opposed to laying an egg, right? Without viruses, you'd all be laying eggs.
That was Professor Vincent Racaniello. Highly recommended. Check him out. Good videos from his lectures talking about how remarkable this case is of the placenta and how an ERV is crucial for the mammalian reproduction of the higher mammalian organisms. Here is a paper entitled Friendly Viruses. So now viruses are friendly because they are enabling these fantastic designs. Independent ERVs contributed to placental morphogenesis. Wow. ERV proteins, quote, have been domesticated independently on multiple occasions for the placental functions of several mammalian lineages, thus representing a process of convergent evolution. Now here's a paper entitled From Junk DNA to Genomic Treasure. ERVs are transformed from junk to essential genomic players. Now this makes no sense under evolution. Another rapidly evolving system with clear signs of ERV involvement is the mammalian placenta, thought to be driven by independent ERV acquisition. These events provide species-specific placental programs in a remarkable example of convergent evolution across all placental mammals. Multiple acquisition events of ERV proteins independently resulted in a remarkable example of convergent evolution. Okay, let's look at another example of ERVs being crucial to an advanced design, the myelin sheath. A recent and remarkable example of convergent evolution was an ERV essential for myelination. What is myelination? What is the myelin sheath? Well, let's read on here. Myelin is the insulating sheath surrounding axons in the central nervous system that allows for rapid transmission of nerve impulses to enable more complex brains. Surprisingly, evidence suggests that multiple independent species-specific ERV integrations are important for myelination. This is further evidence that convergent evolution plays a central role in driving genetic innovation. The myelin sheath. Now here's a nice graphic. On the left you can see this red blob. This is a neuron and its axon tail sticking out. So that's a long tail that sends electrical signals, the action potentials. And like any wire carrying electricity, it has a sheath around it, an insulating sheath. That's the myelin sheath there. Retroviral endogenization played a prominent role in the emergence of vertebrate myelin. This innovation enabled rapid transmission of nerve impulses, more complex brains, and greater morphological diversity. A critical step in vertebrate evolution. Wow, a critical step. So let's look at another example, the human brain, a kind of related example here. This supports the idea that retrotransposon-dependent evolutionary novelties were crucial for the emergence of mammalian, primate, and human peculiarities of brain morphology and function. Recent studies, which are increasingly revealing the pervasive contribution of the different groups of retrotransposons to the evolution of distinguishing brain features in mammals and particularly in humans. Both LTR and non-LTR retrotransposons appear to have contributed to mammalian brain evolution by acting as sources of novel non-coding RNAs, proteins enhancers, RNA regulatory sites, and sites for 3D genome organization. So here's a nice graphic showing these different enhancements, these different innovations. All of these are novel. What are the chances that a virus evolved and then provided all these drivers of human brain evolution? And for no reason. Under evolutionary theory, there can be no reason. So, incredible serendipity, astronomical serendipity off the charts. Evolution creates viruses that just happen to create evolution and create incredible designs. That's serendipity. These are thought to be major agents. Viruses are thought to be major agents of evolution by virtue of their capacity to operate as vehicles of horizontal gene transfer. Yet another mechanism here that viruses are going to perform to create more evolution. Evolution created more evolution. But there is more. Over and over and over again, it is convergent. It's happening over and over and over again. So we're seeing incredible serendipity, but not just once. So here's back to this myelination. Each of these red triangles is where it happens again, independently, convergently. This serendipity is not just once. You can't ascribe it to a common descent pattern. It could not have been via common descent. It must have been via convergence. The syncytians, these are these proteins that are helping with the placental. These syncytians are a striking example of convergent evolution, having originated independently across multiple mammalian lineages, including marsupials, as well as in at least one species of live-bearing reptile. It is noteworthy that the genes that are required for placental development, now this is a paper about the myelin sheath, okay, so they're trying to explain, how could this have happened actually? So what they're saying here is that it is noteworthy that the genes that are required for the placental development came from an ERV via independent events. across diverse mammalian species, as well as one species of lizard, another related example of convergent evolution. So in other words, hey, it happened before, so let's not be too shocked by this. In other words, if it could happen with placental development, then it could happen with the myelin sheath. Again, we've seen this before. This is their way of explaining it. It's an incredible evolution falsifier, but, well, hey, we've seen it before. So no big deal. ERVs actively shape host evolution through complex mechanisms involving genetic innovation. I couldn't have said it better myself. Remarkably, certain ERV components perform critical cellular functions. These now constitute integral components of host genomic architecture and regulation. It is now well established that ERVs constitute integral components of all vertebrate genomes. So I call this convergent serendipity. Here's a new term. I like this term, convergent serendipity. So let's summarize so far what we've seen. Virus evolution is crucial for evolutionary theory, but there is no plausible cause of virus evolution. No one knows how viruses evolved. Evolutionists believe viruses evolve in spite of the evidence. For endogenous retroviruses, there is an LTR proviral mismatch, proviral body mismatch. It contradicts evolution, and it is widespread. If you are an evolutionist, ERVs must create evolution itself and fantastic biological designs. And ERVs must do this repeatedly and independently in a ridiculously astonishing convergence. This is a train wreck. But wait, what about that probability calculation that we saw way back at the beginning of this video? I said I would get to that. Remember, it was one chance in 10 to the 1,418. That's a 1 followed by thousands of zeros. In other words, impossible. There's no way. It is impossible. But what exactly is impossible again?
In contrast, if we want to believe the fixed species view, we're forced to conclude that these viruses are simply shared by coincidence. When we do the math, even making sure to account for the 9 viruses not shared by the 2 species, the chance of this happening by coincidence is less than 1 in this crazy number right here. This evidence should be enough for even the most reluctant yet rational person to carefully set aside the fixed species view.
The fixed species view. That is what is impossible. That's what they are saying is impossible. We are supposed to set aside the fixed species view. And our evolutionist friend agrees. It's astronomically improbable. Very strong evidence for evolution and common ancestry. The video says any rational person should set aside the fixed species view. So the subject is the fixed species view. Nothing about these endogenous retroviruses, or viruses more generally, actually helps evolutionary theory. It is an argument against a different theory. A proof by the process of elimination. You have theories A and B. If I can disprove B, then only A is left. Theory B in this case is creationism, or the fixity of species, which goes back to the 18th century and before. What is being evaluated and tested here is not evolution. It is the fixity of species theory. Evolution is declared to be true by the failure of the fixity of species. That's how this argument is working. So imagine this box contains all possibilities. You have evolution, and you have the fixity of species. If the fixity of species is false, then only evolution remains. Evolution is true by virtue of falsifying the fixity of species. But wait, how do evolutionists know the possibilities? How do evolutionists know there are only 2 possibilities? How do evolutionists know the non-evolution possibility? How do they know there is only 1 non-evolution possibility? And how do they know what it is? What experiment did they perform to learn this? What experiment revealed the possibilities? The answer is, of course, there was none. There was no experiment because this is not a scientific argument. It is a religious argument. So we have bad science driven by religion. Evolution is an absurd theory that must be true. Religion drives science, and it matters.